Yes, friends. I wanted to give you a little lesson on restriction enzymes today. So what restriction enzymes do is I'm going to just do this as if I were doing it on the whiteboard, which is what I would do if we were in class. They are scissors, okay? I want you to think of them as scissors. And what they cut is they cut DNA, okay? Now, when we talk about DNA, we talked about DNA is made up of 3 parts. Remember, we talked about this. Those of you all that came in, you got a special lesson on this from me. DNA has got the 5-carbon sugar. You probably want to take these notes with me. If I'm writing them down, it's probably not a bad idea that you do. A nitrogen base and a phosphate group. So the 5-carbon sugar is deoxyribose, or at least make sure they're in your notes on the notes pages that you took. The 5-carbon sugar is deoxyribose. That's where DNA gets its name, deoxyribonucleic acid. Anything that ends in ose is a sugar, okay? And so that's how we know it's a sugar. It's a 5-carbon sugar, so if you see it, those of you that came in and did the model, it kind of looks like a pentagon, because anytime you see like a shape, when we're talking about a chemical structure, these corners represent a carbon. So if you had a hexagon, that would be some type of 6-carbon sugar, like glucose. Again, it ends in ose, so we know glucose is a sugar. Anything that ends in ose is a sugar. Glucose, fructose, galactose, maltose, sucrose, which is table sugar that you put in your coffee. All of those things, okay? And then our nitrogen bases, those are, if I can write, I got on my eraser somehow. Those are adenine, thymine, guanine, and cytosine, okay? Remember, these 2 pair together and these 2 pair together. That's known as Chargaff's rules. You should have read about Chargaff's rules, okay? And so if we have a strand of DNA is just gonna look like these letters all split out. Okay. And then the other strand, because DNA is a double-stranded sequence, would be the opposite of those letters. So if I gave you one strand of DNA, you should easily be able to come up with the other side based on those Chargaff's rules. A pairs with T, T pairs with A, G pairs with C, A pairs with T, and so on and so forth, using these Chargaff's rules here, okay? So, restriction enzymes, what they do is they recognize a specific sequence of DNA, okay? And so, let's say, for example, we have a restriction enzyme that recognizes the sequence C, C, A, T, C, and it makes its cut down and over, okay? So C, C, T, A, C, down and there, okay? And that's how it cuts. It's going to tell you what it recognizes and what it is cutting between. And so that restriction enzyme, it'll have a special name. They have funny names, like HEN3 is one you hear about a lot. EcoR1 is a restriction enzyme. They have weird names like these. That's not the name of either of this one. But it cuts between this sequence every single time. Now, this is a special type of cut called a sticky cut, okay? And it goes down and goes between the 2 sequences. Now, I just drew this out from my head, so this one didn't work perfectly because I just, like I said, drew it out from my head. But generally what happens is the sequence the same on the top and the bottom. So like this should have been a C, and this should have been a C, and this should have been an A, and this should have been a T. It would have been, it's a palindrome. Like, for example, the name Hannah. If you read Hannah frontways or backways, it's H-A-N-N-A-H. If you read it backwards, it's H-A-N-N-A-H. And so restriction enzymes work in the same way. It's going to read in the same, it's going to be the same frontways or backways. Mine didn't work out that perfectly because, like I said, I just made that up off the top of my head. Okay, but yours in your notebook will work out perfectly. And so it recognizes the sequence and it makes the cut. It tells you what sequence it cuts between. You don't have to memorize those. You don't have to know that it cuts it a certain way. It's going to tell you what sequence it cuts between it. You just have to look for that sequence of letters and draw the cut, okay? Again, this is a sticky cut. Some of them may be a blunt cut. So, for example, maybe a different restriction enzyme recognizes this sequence and it cuts here. And so in this DNA, it would make that cut. That's referred to as a blunt cut, okay? And so a restriction enzyme chops DNA up into smaller pieces. And these pieces that are left are referred to as restriction fragments, okay? These overhangs here that you sometimes get, those are called sticky ends, okay? Now you don't have a sticky end in a blunt cut because it cuts straight through and you don't have this overhanging area, okay? But these are called restriction fragments. And so then what you do once you have restriction fragments is you can run the DNA in a process that's known as gel electrophoresis. Now, I don't remember exactly if that has been talked about yet in your reading that you've done so far, but if it hasn't, it is going to be. So I'm going to go ahead and talk a little bit about it. If it's new, then you're getting a preview to it, and if it's old, then you're getting a review of it. So either way, you're going to have to know it. Whether you've already read about it or you're going to read about it in the next few days, you're going to have to know it. So I'm just going to teach on it right now. So once we take our huge DNA molecule, because DNA is really, really big, and we put these restriction enzymes in it, and we chop it up into little bitty pieces, it cuts them into these fragments, then we can put these fragments in a gel electrophoresis. And so we will do this in class. So in a gel electrophoresis, you have a gel. It literally feels like Jell-O, but a solid Jell-O. And at the top of the gel, you have wells, okay? And you put your fragments, you put your DNA samples in the wells. So we've got sample 1, 2, sample 3, and so on and so forth. Okay, and then maybe we have our mystery. Maybe we would have like Anna Garcia's DNA here, or the DNA that was found at the crime scene. So our samples here would be like our suspects. All right, we have her boyfriend, we have her friend, we have her boss, we have all those different people that we've read and talked about. And then we have Anna here. And we're trying to look, or not Anna, I'm sorry, the crime scene. The crime scene DNA here. And we're trying to see where this DNA came from. Did it come from Anna? Did it come from one of the suspects? And we would put Anna maybe in one of these boxes too, because we want to know if it's actually her DNA or not. And we would run a gel electrophoresis on it. And a gel electrophoresis is how you determine whether the DNA is theirs or not. What you get with a gel electrophoresis is you get a DNA profile, or sometimes it's referred to as a DNA fingerprint. Okay, and this is how they do it in the crime shows. When you see that, you know, they match the DNA and they got a such and such % match, this is what they're doing. They are doing gel electrophoresis. They're not explaining it in all the fancy words. They're not talking about the restriction enzymes, but that is what they are doing, and that is what you're learning about. So this is really important. So they load the DNA samples in the wells, and then they put the DNA, gel into a box, okay? And so this is called the electrophoresis chamber. Okay? And you hook it up to electricity. So it's got these cords that come out of it, and you're going to hook it up to electricity. One is going to be hooked to a positive end, and one is going to be hooked to a negative end. And so when you lay this well in here, when you lay your DNA gel electrophoresis in the electrophoresis chamber, you want your wells to be on the negative end, okay? Because DNA has a negative charge. And so if you know anything about opposites, you know that opposites attract. And we want our gel to move this way. It'll pull this negatively charged DNA down to the positive end, okay? And so what you'll get in your gel electrophoresis, after you've run it, it just runs for like 30 or so minutes, and we have this equipment here in the classroom to be able to do this. So we would have suspect 1, suspect 2, suspect 3, let's say Anna, and then let's say our crime scene data. You get a banding pattern that shows how the restriction enzymes separated out. Oh shoot, I didn't do that right. Okay, give me just a second. And so what you should be able to look at here and see is that the crime scene DNA most closely matches DNA of suspect 1. So whoever was suspect 1, whoever's DNA you put in that chamber, that's the person that we have their DNA at the crime scene, whether that was from the blood or from the hair or from a fingerprint or whatever you found. That's the DNA that was found at the crime scene, okay? And you can go, and we have evidence for that now, okay? So I just wanted to give a little bit of background about gel electrophoresis, a little bit of background about restriction enzymes, and then you have your activities for today to be able to work with the restriction enzymes and cut those pieces just like I described to you up here, okay? Let me know if you'll have any questions on it, and I will see you soon for our lab on Friday. Bye.